Hello dear students, I welcome you all to the video tutorial sessions on carbohydrate chemistry under the aegis of OneMedicine. Myself, Dr. Sanghamitra Chakraborty will be the guide by your side for this detailed discussions on carbohydrate chemistry. This is the first video tutorial of carbohydrate chemistry. At the end of this video, you will be able to define the carbohydrates, classify carbohydrates and enumerate various physiological as well as pharmacological actions of carbohydrates. Now, what are carbohydrates? By definition, initially they were defined by the general formula CnH2ON, but it has been seen that this formula do not satisfy all the properties or criteria of carbohydrate. Even some organic compounds like acetic acid having the general formula CH3COOH, it is not a carbohydrate, but it suffices this formula. This formula is sufficed by acetic acid too. After that, the definition of carbohydrate change to polyhydroxyaldehydes or ketone derivatives or compounds which yield this on hydrolysis. So this is the standard definition of carbohydrates. That means they have two functional groups. Either they have the aldehydes or they have the ketone group as the functional group. So this is the diagram of an ketone group and this is the diagram of an aldehyde group. Now let us check the definition, that is polyhydroxyaldehyde or ketone derivatives or compounds which yield this on hydrolysis. All the properties of carbohydrate are basically of either because of this aldehyde group or ketone group. Whenever you are asked in theory exam, define carbohydrates, we know any definition is incomplete without example. So you have to give example like glucose, fructose, sucrose, lactose, mannose, so on and so forth. Clear? Now, classification of carbohydrate. Initially, carbohydrate has its etymological origin from the Greek word saccharon, meaning something sweet to taste, or sugar. So depending on the sugar units, carbohydrates are classified as monosaccharide, disaccharides or oligosaccharides and polysaccharides. These are the four types of carbohydrates. Clear? Mono, di, oligo and polysaccharides. Mono means single unit of sugar. One. The simplest form of sugar is monosaccharide. That means it cannot be further hydrolyzed. Now, this monosaccharides, if it has aldehyde as the functional group, they are termed as aldose. If they have ketone group as the functional group, then they are termed as ketose. So, they are basically of two types, aldose sugar or ketose sugar. Depending on the number of carbon atoms, these are the common monosaccharides which are having some physiological importance in our body. To start with, the glyceraldehyde and its complementary ketose sugar is dihydroxyacetone. It is the 3 carbon containing sugar. Next is erythrose. Its complementary is erythrolose, 4 carbon. Ribose, ribolose, 5 carbon. Glucose and fructose, we all know 6 carbon containing sugar. They are the hexoses. And glucoheptose, 7 carbon containing aldose sugar. Pseudoheptulose, 7 carbon containing ketose sugar. Now, how their naming is done? For aldose, it is OSE at the suffix, and for ketose, it is OLUSE at the suffix. So, OS means aldose, OLOS means ketose. This is for your idea how they are named. And these are important. When we will look in the detailed discussion of the isomerism of the monosaccharides, their structural property, we will see that this glyceraldehyde or this dihydroxyacetone has been used as the parent compound. This is the parent compound depending on which the structural isomerism of monosaccharides are done. Next, after mono, di. That means 2 sugar units, disaccharides. They can be divided into reducing or non-reducing. That means Here in disaccharides, the two monosaccharides are linked by glycosidic linkage. Now, if they have the free carbonyl group, that is free ketone or aldehyde carbonyl oxygen is free, then they will show reducing property. If both of them are utilized in the glycosidic linkage, they will not show reducing property and they are termed as non-reducing sugar. Now, this disaccharides reducing like lactose, which is having galactose plus glucose, and maltose, which are having two identical glucose units. These are reducing disaccharide. Lactose is the milk sugar. Non-reducing is sucrose, the cane sugar, which consists of glucose and fructose and trehalose. In exam, a common question is, give some example of non-reducing sugar. One is sucrose, second one is trehalose. You have to remember these names. Next, we are moving on to third type of carbohydrate, that is oligosaccharides. Oligosaccharides, they have 3 to 10 units of sugar. They are known as oligosaccharides. They do not occur as free entities. They occur as glycoconjugates. That means they occur as glycoprotein or glycolipids. Glycoprotein or glycolipids. Now, these oligosaccharides, they are again termed as some of them are free and they are taken from plant sources. They have their existence in plant sources, free oligosaccharides. Let us check them. First one is the raffinose. This raffinose, it is a trisaccharide. This raffinose, it is a trisaccharide. It consists of glucose, galactose and fructose. Stachyose, it is a tetrasaccharide. It consists of 2 galactose unit, 1 glucose unit and 1 fructose unit. Verbascose, it is a pentasaccharide. It consists of 3 galactose, 1 glucose and 1 fructose unit. So oligosaccharides, they do not occur as free entities in our physiology. They occur as glycoconjugates in glycoproteins or glycolipids. But in plant, they are there. Like raffinose, they are found in broccoli, cauliflowers. Stachyose are also found in plants, and in verbascose, they are found in legumes and lentils, and they causes flatulence. So these are the oligosaccharides. Next, moving on to the fourth type of carbohydrate, that is polysaccharide. Polysaccharides, they have more than 10 units of sugar. They are polysaccharides. If they are linked by the same type of sugar units repeatedly, they are known as homopolysaccharides. If one glucose is linked with another, like this, more than 10 units of glucose are there, for an example, they are called as homopolysaccharide. And if they are linked with different sugar units, they are termed as heteropolysaccharide. Homopolysaccharides are also called as homoglycans, and like heteropolysaccharides are also called as heteroglycans. So these are the types of homopolysaccharides and heteropolysaccharides. Now let us check them. Homopolysaccharides, the examples are starch, the reserved carbohydrate in plant kingdom, glycogen, cellulose, inulin, dextran, chitin and dextrin. Heteroglycans are agar, agarose, gum and pectin, hyaluronic acid, chondroitin sulfate, keratan sulfate, darmatan sulfate and heparan sulfate. This, all of them, they are coded as another color. If you look at this table, this last 5, they are coded as another color because they are also known as mucopolysaccharides. They are also known as mucopolysaccharides because they have been first isolated from the mucin, and that's why they are termed as mucopolysaccharides. So we have an idea of the classification. Monosaccharide, single unit of sugar, further no hydrolysis, simplest, glucose, disaccharide, depending on the utilization of the carbonyl group, reducing and non-reducing, reducing lactose, non-reducing sucrose. Oligosaccharides 3 to 10. In our physiology, they occur as glycoconjugates, no free existence, but in plant, they have existence like raffinose, stachyose and verbascose. Polysaccharide, more than 10 units, homoglycans, heteroglycan, homoglycan, starch, glycogen and cellulose, heteroglycan, agar, agarose, pectin, heparan sulfate, keratan sulfate, chondroitin sulfate, darmatan sulfate, hyaluronic acid, they are all heteroglycans. So this is an idea of the classification of carbohydrates. Now, uses of carbohydrates. The physiological uses of carbohydrates. Let us check them first. These carbohydrates, they are the dietary source of energy. They serve as the dietary source of energy. 1 gram of carbohydrate, if it is oxidized completely, it will yield 4 kilocalorie of energy. 1 gram of carbohydrate. Maximum bulk of energy in our food that is contributed by the carbohydrate. Reserved from glycogen. If we are fasting, after 8 to 10 hours of fasting, the glycogenolysis will start, and after 12 to 18 hours of complete fasting, the entire glycogen reserve in our body will be exhausted. They are stored in the liver and the muscle. They act as dietary fibers, that means they form the bulk or the roughage part of the diet, and they like cellulose, pectin, lignins, they form the fiber constituent of the diet. They occur as glycoconjugate and they form precursors of glycoproteins and glycolipids like N-acetyl glucosamine, N-acetyl galactosamine, and they are important constituent of the cell membrane that help in cell-to-cell interaction or cell signaling. And they also are also involved in detoxification reaction. They are also involved in detoxification reaction. That means when we will study the metabolism of carbohydrate, we will see that glucose can be oxidized to glucuronic acid, and this glucuronic acid is used for detoxification of bilirubin, for detoxification of steroid hormones. Like this, they are also serving as a detoxifying agent. After physiological function, we will move on to pharmacological actions of the carbohydrate. Now, first one is dextrose infusion. You go to the clinics, you will see majority of the patients who are on NPM, that is nothing per mouth, they are given this dextrose infusion. This is nothing but glucose infusion to maintain the homeostasis of the glucose in the physiology. They are given glucose infusion. Next, this is the bottle of a glucose. Move on to the word. You can see this is stacked in the words. Mannitol. Next, coming to mannitol. What is this mannitol? This mannitol is nothing but it is the alcohol derivative of mannose. Recall the definition: polyhydroxyaldehyde or ketone derivatives or compounds which on hydrolysis give this one. So this mannitol is derived from mannose and it is used in lowering of intracranial tension in stroke. It is given to the patients. Lactulose. This lactulose is used in the treatment of hepatic encephalopathy. This lactulose is used in the treatment of hepatic encephalopathy. And last one is the inulin. It is a homopolysaccharide of fructose. It is extracted from dahlia tubers and it is used as a gold standard for estimation of glomerular filtration rate. So these are the pharmacological uses of glucose. To recapitulate, carbohydrates, polyhydroxyaldehydes or ketones, derivatives or compounds which on hydrolysis yield this. Example: glucose, fructose, sucrose. They are classified on the number of sugar units as monosaccharide, disaccharides, oligosaccharides and polysaccharides. Mono, single, di, 2, oligo, 3 to 10, poly, more than 10, poly again, homoglycans and heteroglycans. And these are the physiological and pharmacological functions of the carbohydrates. From the next sessions, we will move to the detailed chemistry of each of the subtypes of the carbohydrate.